Hi, this is Gary Petersen, and welcome to CureTalks with Gary. We'll be talking about new breakthrough research in therapies with CEOs and senior level management of companies. The first of this series is next generation targeted cancer therapies with Cellectar Pharmaceuticals. So we welcome you all. Small biotech has been the genesis of every approved class of drugs for myeloma, and all new myeloma classes of drugs have had at least 2 FDA designations, like Orphan Drug or Fast Track. Specifically, that just means that the FDA has looked at them, feels that these drugs are really anointed as great drugs for these orphan diseases. Myeloma, for example, has 8 such drugs with dual designations, and 6 of those are from small pharma. So small pharma, like Cellectar, has 4 such designations for orphan diseases and is a new class of drug with application across many cancers. I am so pleased to be able to discuss this life-saving development of Cellectar with senior level management. We are honored to have Cellectar CEO and President James Caruso, as well as the Chief Business Officer Jared Longcore. So welcome to both of you. I'd like to break this into 2 parts, the first of which is just a general outline of Cellectar, and I'd like to do that with James Caruso. James, would you please provide a background and history of the company and how your organization can do so much with so little?
Well, it's my pleasure, Gary. And first and foremost, thank you very much for allowing us to participate in your program. You do great work, and I'm sure it's very rewarding for you and is very helpful for your audience. So we're very appreciative of having the opportunity to be here with you today. Cellectar Biosciences is a very interesting story. I mean, we're located in Florham Park, New Jersey. However, the company originated in the Midwest, and the technology, our base technology, that Jared will talk to in greater detail, and I'm sure your audience is very interested in learning more about our delivery platform and the targeted mechanism that makes it our proprietary technology unique and different. This technology came from the University of Michigan, and then researchers and academics transitioned to the University of Madison, Wisconsin. And that's where Cellectar Biosciences was founded and originated. Interestingly, this technology, this targeted mechanism, if you will, was designed for the delivery of diagnostic and optical imaging agents. So with the delivery vehicle, these researchers and scientists were adding fluorescent imaging agents and diagnostic agents for PET scan, et cetera. And the delivery vehicle, time and time again, regardless of the types of payloads added, demonstrated, a preferential delivery to cancer cells, cancer stem cells, and metastases, sparing healthy tissue. Now, the company was doing some great research, but unfortunately it was failing financially. And as a result, was close to shutting its doors and going bankrupt. When Jared and I first came to the company, we took a real hard look at the technology. And what we realized was this technology could better serve cancer patients and better deliver on the growth and success of the company if it was redeployed, not a focus on diagnostic and optical imaging agents, but said another way, let's take oncology therapeutic payloads, add them to this delivery vehicle, and now we can treat cancer patients. And so the company at the time, Gary, had very little money, as I mentioned, was going bankrupt, and we were able to cobble together some financing and some capital, and we were able to reposition the company very quickly, and we pivoted towards the treatment of cancer. And we started in the heme malignancy space, if you will. And we wanted to focus on an area that we knew we could accelerate through the clinic and really help patients. And that was in these orphan and ultra orphan spaces. That really made a lot of sense for us on a lot of different levels. And, fast forward 5 years later, that's where the company currently sits. I mean, we've survived over those 5 years. It's been a very challenging journey, but we're really now on the cusp of transformation, most importantly for patients, as we launch into a pivotal trial in the fourth quarter of this year. As Jared will talk to, our data looks really, really good. And it looks really good in very difficult to treat patient populations. So we have the opportunity to do some really good with CLR 131, which is the lead asset. Now, you also mentioned, and I think this is important, with your P&L, the size of your P&L, how has the team, done so much with so little? And I will say, just like anything, Gary, first and foremost, it's about your people. We just have a great team here at Cellectar, highly dedicated and highly committed in our battle against cancer. Most experiences that fuel, either with family or friends, that fuel this dedication and this commitment. And as an organization, our low level of productivity is very high. And part of the reason for that is, we work double time in terms of working on CLR 131, getting this through the clinic and hopefully providing a compound or a drug that can help patients. Now, on the functional side of that, we've installed what we call a collaborative kind of outsourcing model, if you will. And what this outsourcing model has allowed us to do, it's really kind of limit our infrastructure and the day-to-day costs of running the company. And we've outsourced through quality third-party vendors a lot of work. And what this allows us to do, Gary, is much, much more with much, much less, because we don't sit on this heavy infrastructure and day-to-day cost. What the collaborative outsourcing model has allowed us to do is really invest our limited capital and resources in projects and areas that we believe can really advance our research. So as I mentioned, it's a small team, but we're highly productive. We manage the P&L very, very tightly. It's as thin of a P&L statement as you're going to see. And thanks to the team, we get significant bang for a dollar.
Well, it seems, and you might expound on that, what are the additional challenges that confront a small biotech company versus a huge biotech company like Johnson & Johnson, Pfizer, or others such as that? What are the specific challenges you have? One of which is, like you mentioned, just putting together, the resources in order to move things forward.
No, that's a great question, and it's a fair question as well. I mean, I've had the opportunity to work both in large multinational pharmaceutical companies as well as in 3 or 4 small biotech companies as well. So I'm familiar with both business models, and they're significantly different, right? I mean, in these larger companies, access to resources is obviously not an issue. They maintain significant infrastructure. They can sustain significant investment in research and development and maintain, 30, 40, 50,000 employees as a result. For smaller biotech companies, and, we have, we're low double digits. We have to do a lot more with a lot less. And for us, it's access to capital and financing, which is why when when we're very thoughtful and strategic in terms of our investments and finances, we then have to take that money and be so judicious in terms of how we invest that capital to ensure that we can optimize those resources and advance our clinical development as far as we possibly can. I think what's also really interesting, because of the large scale of the multinational pharmaceutical companies, there's a lot of mistakes that are made, and they're transparent to the public because they're so big and have so many resources. Where in smaller companies, we have to be razor sharp day in and day out because we can't afford to make a mistake. right? Because these mistakes, unfortunately, or mishaps, could have an impact into both the near term and the future term. So, we work without a safety net, which makes it very, very challenging. And so that's why you need a team that's totally dedicated and committed, to get these types of projects and this type of work done and completed. I would say the other, there's a lot of differences here, but the other one that I think your audience would be appreciative of is just the scale and the size of the infrastructure associated with these large multinational pharmaceutical companies, right? So they have dozens and dozens of developed products in development, typically dozens of products that are commercialized on market, generating revenue. And if one or two or three or four or five or six fail in the clinic, it's really at the end of the day non-material, right? Because they can backfill with additional products that are in development. And the same is true commercially. Sometimes there's forecasts, and if those forecasts are not reached for a particular asset or compound, there's dozens of others that can fill that gap. With smaller biotech companies, you typically don't have this massive R&D budget, and you typically don't have dozens of products or any product on the market. And so your revenue is generated through financing. What we've done to try to distinguish Cellectar Biosciences from other like small biotechs our size is we've really tried to mitigate risk. And said another way, we've kind of spread the field here with CLR 131, our lead asset. And what we've done is we focused in on multiple B-cell malignancies, almost like a basket approach, and have had really nice data in multiple myeloma subsets. We've had really nice data in Waldenstrom's, another ultra-orphan disease, as well as DLBCL and other NHL and non-Hodgkin's lymphoma cancers or indications. And so what we wanted to do, Gary, was not focus just on one indication, where if we were unsuccessful there, patients potentially may not benefit from what we believe are real feature benefits of CLR 131. There's really some unique product profile elements that make this compound unique versus on-market existing products. But we also wanted to create opportunity where if we were unsuccessful in one area, we had multiple other indications that we could potentially bring this product to market.
Yeah, I've got one more question I'd like to ask before we move on. I want to give Jared an opportunity to discuss the product a little bit as well. But one of the things that I've seen, and you were talking about it, is that just being able to fund the company and how you do that. And I've had my eye on looking at some of the things that happen in the capital markets, and it's kind of tough to I've made people's eyes glaze over because when I talk about it. But if you can talk to the audience without getting them all glassy-eyed about how does uncontrolled predatory stock short sales affect your company's ability to expedite product development. Obviously, you could do more if you had more.
No, absolutely, Gary. And one of the things I do best is have eyes gloss over. So I'll be pretty direct of responding to this. I think for small cap companies in general, you're spot on, right? You're really subject to the markets. Your stock price is really your currency. When you don't have commercial revenue, you fund the clinical development of your assets, through the sale of your stock. And so to your point, in the small cap space, you could have individuals that may short the stock, and those types of activities may have a negative impact on the growth of your stock. And it is a significant issue. At Cellectar, on the blocking, enhancing the clinical development of CLR 131, because we know if we can deliver a product to market that can help patients, that will transform the company and also potentially transform the life of that patient, their families, and potentially the communities that they live in. Well,
I know soon you're going to probably have a phase 3 trial for Cellectar. And based on my research, if you get into a phase 3 trial with an orphan drug, you have at least a 75% chance of FDA approval. So to me, I just do not understand, like your stock is valued much less now than it was at your initial IPO. However, you're closer to the finish line than ever. So, that may, you're on the 2-yard line, you're about to put the ball over, and people still don't see that, and it just blows my mind. It's mathematics, and a little bit of probability theory. But in any event, that I appreciate you and all the things that you do and all the things that small biotechs, because without small biotech, without IMiDs, without proteasome inhibitors, without monoclonal antibodies, I'd be dead. So thank you. Now I'd like to move on right now, if you don't mind, and I'd like to go on to Jared. Jared, I'm just so happy to see you again. I saw you at ASH and I stopped by, and to your point, James, is that I talked with both Jared, and I can't remember the other person, but she was involved mostly with the presentations, I guess, at at ASH, and they were just stellar people. So you do have a great team behind you, and my conversations now with you just shows they've got great leadership as well. So Jared, I'm intrigued by the mechanism of action for CLL-131. And can you explain what that action is specifically?
Absolutely, Gary. It'd be my pleasure. So I'm going to actually cause everybody's eyes to glaze over, because that's what I do best when I talk about science. I'm very talented at that. So I'm going to try and simplify this as much as I possibly can to keep it sort of not too technical. But simply speaking, the way you can think about CLR-131 is essentially like an antibody drug conjugate. We've got a radioisotope attached to one side of it, and then we specifically target to the tumor cells with our proprietary delivery vehicle known as a phospholipid ether. Now, how does that actually work? How do we successfully target using a phospholipid to a tumor cell? As many people in your audience may know, tumor cells actually have a significant metabolic need. These needs are increased due to the division of the cell. They have increased need for energy, so ATP, and they have increased need for signaling molecules besides, having the phospholipid membrane increase. In that auspices, all cells, when they make ATP, have sort of two mechanisms that they use. Normal cells and cancer cells use the same two mechanisms. One is called glycolysis, or the breakdown of glucose to create ATP, and the other is called the beta oxidative pathway, which is the breakdown of long-chain fatty acids and phospholipids to create ATP. In normal tissue, that ratio of glucose or glycolysis to beta oxidative pathway is like 80 to 90% on glycolysis and 10 to 20% on the beta oxidative pathway. In tumor cells, that shifts and it becomes more of a 50-50 to 40-60. So there's a lot more utilization of the beta oxidative pathway. In addition to that, the tumor cells need for those additional signaling molecules and for extra lipid membrane potential requires them to actually modify their cell surface. That modification allows them to actually pull in these molecules out of the bloodstream, these phospholipids and long-chain fatty acids right out of the bloodstream. And then the way they do this is they create specialized localized regions on the cell membrane known as lipid rafts. And these lipid rafts form and they exist for upwards of 10 days. And that's exactly what we bind to. So when you think of an antibody drug conjugate, when they bind one antibody to one epitope, what we do is we bind a lot of molecules to a single lipid raft. And these lipid rafts, it's not like there's just one on the cells, on the tumor cell surface. There are hundreds. And what we've seen is that's universal across many of these tumor cells. So that's about the simplest way I can describe it.
Okay. Now, you said normal cells also have that, but in less quantity, right? So are they also affected?
Yeah, so normal cells do have it, but what changes is in a normal cell, these lipid rafts will form and dissipate in about 2 nanoseconds. So very short period of time. And then they are in very low copy numbers. So in the orders of 10 to 20 sort of lipid rafts on the cell surface at any time. When you look at a tumor cell, as I mentioned, these lipid rafts are stabilized for up to 10 days. So they exist there. One will exist there for 7 to 10 days on average. And then what you see is that the copy number or the amount of lipid rafts on the cell surface are more in the orders of hundreds to thousands as compared to 10 to 20. And so that's sort of the change, and that's what gives us the ability to get so much more drug into the tumor cells and get so much more targeting out of this as opposed to some other technologies.
So it's like battleship. The more battleships you have out there, the more likely you are to hit one.
There you go. That's a good analogy. I used to always tell people, it's sort of, we've sort of engineered probably, people used to talk about the Trojan horse approach with cancer. And I think this is about as close to a true Trojan horse. The tumor cells can't help. They have to take up these long chain fatty acids. And what we have is essentially a long chain fatty acid mimetic. And the tumor cells suck us in like a vacuum cleaner, not knowing what we're bringing with us. And once we're in, then we do our business and we kill the tumor cell from within.
Okay.
And that's the real secret.
One of the newest drugs available for blood cancer is a monoclonal antibody. But, this has to target a specific antigen like CD38, BCMA. And why is your method of targeting cancer not require a specific antigen? And I think you kind of said that, but I think an adjunct to that would be, do all cancer cells, why yours seems to go across boundaries. So do all cancer cells have this same need of for the rafts and for us to sink those little suckers?
Like Battleship? Yes.
Yeah.
So what I'd say is, go ahead.
Okay, no, go.
Okay. So what I'd say is, the reality is, you said it before with regard to antibodies and the benefits that patients see. But when you look at an antibody comparing to a phospholipid ether, antibodies, as you said, target a specific antigen. And let's take CD38. CD38 is expressed on multiple myeloma cells, but it's not expressed on all of the cells, and it's not overexpressed in 100% of the patients. In fact, if you look at daratumumab, which is an anti-CD38 antibody, it works in about 30% of the patients. Why? Because the antigen, the CD38, is overexpressed in about 30% of the patients. And inside that 30% of patients, it may be overexpressed only on 50% of the tumor cells that are actually in that patient. So you never get to 100% of the cells being targeted. To your question, when it comes to lipid rafts, in every tumor type we've looked, in every type of tumor arena we've looked, we see that the lipid rafts are conserved, whether that be in cancer stem cells, whether that be in the primary tumor or in metastatic tissue. We've been able to find, that not only lipid rafts present, but they're overexpressed at a high abundance. Now that's always there, and there's always plenty of them. And this is what we see. We've been able to target cells, what we call circulating tumor cells. These are cells that get into tumor cells that get into the bloodstream or actually can see new metastases. We're actually able to target those. We're able to target what's called micrometastases, so a small, very small collection of tumor cells, often as in a different body part. And so what we're seeing is that we can cover a lot of tumor goes through, as I call it, its life cycle. So from a cancer stem cell to a primary tissue to a metastatic tissue, those antigens that are expressed along that way change over the course of time. This is why what you see is patients develop resistance to treatments, because what you're targeting changes and mutates. The cancer is trying to survive despite the challenges you're giving it. In the case of these lipid rafts, they never get rid of them. They're constitutively present because of this need for the energy source. And that's what we're taking advantage of, how we're getting in.
So you're cutting off the electricity.
Basically, eventually that's what we end up doing. We shut them down.
Okay, so many new drugs have been approved, like selinexor, daratumumab, elotuzumab, all of which as a single agent really don't have much activity whatsoever. Yet fractional dosing for CLR 131, which is, I guess you give it twice over a period of time, has a 50% response rate in heavily pretreated patients. Now, to me, that's remarkable. And why is this such a secret to the myeloma patient and for all of oncology for that matter? Is it another example of size matters?
Yeah, a great question. I think size does make a difference to some extent here. Let's start with the first one you mentioned there, Cellectar and Karyopharm. I think if we look back at when Karyopharm was first getting going, and they were in phase 1 and early phase 2 studies, they might not have been as well known or understood and Cellectar, or the compound itself, may not have been widely known. I think as they got later in that development into late phase 2, into their pivotal study, phase 3 study, then they garnered more awareness, and they also, at that point, they became a larger company. To your question to Jim before, they got to a point where their market cap was bigger, and they were starting now running 2-3 studies at a time. When you go to, and that's where size really matters, and this goes to what Jim was saying earlier, when you look at a large pharma like a J&J, this is where size really does matter. When they're developing daratumumab, they're not running one study in multiple myeloma. They're running a half dozen or dozen studies, looking at it in combination, looking at it in a different dosing paradigm, looking at it in subpopulations in a separate sub-study. What does that do? That increases their awareness across not only the key opinion leaders and the clinicians out there, but also patients hear more about it because they come across it in all these studies and they get a better sense of what's going on. In our case, as Jim mentioned, we're a small company. We have a very small P&L, very targeted P&L. We have to know where we're going, and we have to be very judicious and targeted in our studies because we just don't have $1 billion to spend today to spread out like that. And I think that's where the awareness comes down. I think right now, we're putting out that data, that phase 2 data that you're referring to, with nearly a 50% response rate in, approximately, coming out of 53 patients across 2 dosing groups. That's meaningful data. And I think that starts to raise our awareness, and people are going to start and have started to take notice of Cellectar and CLR 131. And I think that's going to increase now as we continue down the road.
Well, thank you. For many solid tumors, the best treatments has historically been to cut it out, because that kind of gets rid of it if it's a solid tumor, and then to radiate it to kill the remaining tumor cells around where the cancer was growing. Now, however, your drug carries a radioisotope which will circulate in the blood. And I know literature says it's safe, but I'm wondering, how do you ensure that the patient will not turn into the next Spider-Man?
If I could, I would. I mean, it'd be really fun. Nothing wrong with being Spider-Man in my mind. But no, joking aside, and I will agree with you. Yes, the literature does say that targeted radiotherapeutics of this type, whether you call them immunoconjugates or what have you, are very safe. They've been used for a long time, several decades, with some of the more recent ones. And there's older literature that talks about early days with radiotherapeutics being injected. The secret with our drug, though, with CLR 131, is that uptake. How much drug do we actually get to the tumor? When you use an antibody, less than 1% of the antibody that is, let me say that differently. When you infuse the drug, less than 1% of that amount of drug that's infused into the patient actually gets into the tumor. In our case, we see up to 40% of our infused drug in the tumor at the end of the day. Why does that happen? We accumulate into the tumor cell over the course of a very short window. We start at within 5 minutes of infusion, you can start to see CLR 131 or any one of our preclinical programs. You can see those molecules start to accumulate into the tumor cells in that very early window. And that continues over the next course of days. What happens is now you've taken, to your point, if you take 100% of what we injected into the patient, 40% of it gets into the tumor. That only leaves you maybe 60% still circulating for a short period of time, and then that's excreted relatively quickly. So there's a relatively low probability, low, what do I want to say? There's a low amount circulating in the patient at any given time. In addition to that, and I know I'm going to go on a little bit here. In addition to that, we actually inject a very low dose in comparison to other treatments. If you look at something like what is used in the pediatric arena, it's a compound called MIBG 131, which is iodine 131 attached to a different delivery modality. We can deliver 16 times the amount of drug to the tumor that drug delivers. That means we give 16 times less to get the same results. So where they're giving 1,000 millicurie dose, we can give a 80 millicurie dose and get the same results. And that difference means there's much less radiation impacting any of the body. The real beauty in this is when you move beyond just what I'm saying, but you look at our clinical data and our safety profile, that's where you really see this play out. We don't see what's called off-target effects. We're not damaging the healthy tissue when we're circulating. We don't see any changes in the liver enzymes that you monitor for liver impact. You don't see any kidney damage being caused by it, no heart damage, no cardiovascular, nothing in the lungs, nothing in the bloodstream. What you do see is on target. As you mentioned before, you're a multiple myeloma sufferer. In multiple myeloma, where do we go? We go into the bone marrow to get after the tumor cells. We get in there, we get into the tumor cells. What do we see? We see cytopenias or decreases in certain blood counts associated with the drug. That's an on-target effect because our drug is getting right in there at the tumor in the bone marrow, and it will be right next to potentially stem cells that will produce your thrombocytes, your erythrocytes, those cell types in your bloodstream, and they'll be depleted a little bit. And what we've seen is that at any dose we've given, that's all we see. And we don't see, there's been no dose-limiting toxicity associated with those at this point.
Well, fantastic. I appreciate that. My next question had to do with, how it differs from antibody drug conjugates, but you've already talked a little bit about that, unless you want to expound on that at all.
Yeah,
right now, that happens to be one of the big things. I mean, 5 years ago, there was maybe 3 clinical trials at ASH for antibody drug conjugates, and it's exploded. It's like 150 or 200 now. So, I mean, it's just exploded. And it sounds to me like you've got something, that has shown to be as effective or far more effective than some of those that have come out. And yet, so I'm just wondering what the difference is and what makes yours so much more effective, yet invisible to a lot of people.
I think it's, again, that the idea is we get so much more drug into the tumor than an antibody drug conjugate can. When you do an antibody drug conjugate, and this is sort of this will really get into some of the secret sauce of antibody drug conjugates. So remembering that the antibody binds to a single epitope, it's a 1-to-1 ratio, antibody to epitope. This is why antibody drug conjugates, they try to attach 4 or 8 the molecule that's actually doing the work to that antibody. They attach a larger number to it to get more drug into the tumor cell at any given time. Because of that 1-to-1 ratio, it's the only way they can increase the concentration. The greatest challenge with antibody drug conjugates is once they get into the tumor cell, they are taken into what's called a lysosome. It's a subcompartment inside the tumor cell that the drug then has to get back out of and then get to what you want to target. The beauty with what we do is, again, when we bind to the lipid raft, it's not a 1-to-1 ratio. We can put 50 molecules or 100 molecules on any lipid raft and get internalized as a large group. This is why we see so much higher delivery. On the other side of that, we don't go to the lysosome. Because of the mechanism by which we enter the cell, we go directly into the cytoplasm. So we don't have to get outside of another compartment to do what we need to do. We're directly where we want to be. And the way we track inside the cell is we go right to where the nucleus is, in and around the nuclear area. This allows us to, with a radioisotope, that's where you want to be because you want to deliver it as close to the DNA as possible to break it apart. which is why we're so different than some of these other small molecule plays. But when we attach small molecules, it allows us to have greater control over what we attach, how we release it, and that we can bring things into the tumor cell that until now could never have been used in an antibody drug conjugate play. So it's, I wouldn't say, we have competitive advantages over certain things with antibody drug conjugates, but each of these has a place in the treatment paradigm for patients because certain things will work better in certain places.
Yeah, can you please provide us with what you feel is the future of your platform in cancer therapy?
Yeah.
For example, moving it further, what we've always seen is that when you move it, instead of end stage, and we appreciate that. We all, myeloma patients, when we have tried everything, we would like to think that there's one more thing. We're so happy that Cellectar has been approved because it gives us another chance at life. I see CLR 131, the same thing, but more effective, and so you give us another last chance at life. But we also know that when you move these drugs to earlier in the course of the disease, look at daratumumab, had almost no single agent activity, but you move it further back into the when more towards newly diagnosed, it really works. So do you see, where do you see your drug going?
Yeah, no, absolutely. I actually see us sort of straddling in my mind. And what do I mean by that? We can work in the late stage disease setting as you described and be in that more challenging to treat patient population. But in addition to that, I think you could easily see this compound, because of the limited adverse events that we do see with it, will pair up nicely in combination, which is what they're commonly using these days in early lines of treatment, first line, second line, triplets, doublets, some people talking about quadlips, or however you want to say that, quadruple therapies at once. But looking at that paradigm, I can easily see us combined with some of the compounds that are already approved. We would work very synergistically with some of those with CLR 131. But the other nice part about our drug, it's very, some diseases can develop some radio resistance. But if you can get enough radioisotope into those tumor cells, you're still going to break up enough DNA to kill the tumor. And so if a patient takes CLR 131 in first line in combination with something, and then they progress, they could still come back to using CLR 131 in seventh line and still have an effective, meaningful benefit from the drug in both ends of that. And that's why I think of it sort of as straddling. So in multiple myeloma, you can see that, in the case where Jim mentioned earlier, Waldenstrom's macroglobulinemia, a closely related disease to multiple myeloma, we are looking in that space. We have wonderful response rates there, 100% response rate in that patient population. Right now, we're focused in the later line approval process, but again, I see us being utilized in early lines with that. I think you mentioned sort of in the broad scheme of things, we've also got this drug CLR 131 we're developing for pediatric indications.
Several of those.
Yeah, pediatric brain tumors, a host of soft tissue sarcomas, and then in
Talk about save the children.
Yep. Nope. And it's what we see, and we were sharing some interesting information earlier today on this. What we see is exactly what we've been seeing in multiple myeloma. We're seeing that we track to the tumor, we get after it, and it's about getting to the right doses and being able to help the children.
Well, that's outstanding. Obviously I've got my own focus, and happens to be like blood cancers, but the things that I think it's of childhood cancers, 72% of those happen to be rare cancers. So, and nobody wants to work on them because they are so rare. And so I'm just so pleased to hear that you guys are performing in that space, and you have so many drugs for so many of those pediatric cancer. So thank you so much. Well, that was my last question. And just thank you so much for being part of this new series. To me, I don't know why you're such a big secret. I mean, you guys are providing, a potentially game changer when you think a lot of drugs that have been approved for late-line treatment are 5, 10, 25, 30% at a high end, and you guys are at 50% on your fractional dosing is just plain remarkable. So James and Jared, thank you so much for all that you do for and the fight that you guys put up, just to stay in business and to deliver these things, because you guys are really the ones that can save life. You've done it for millions of myeloma patients. The small biotech companies, because not one drug, Velcade was a little small company in the very beginning before it got bought up by Takeda. Cellectar was a $10 million company losing bucks left and right when they picked up thalidomide, which was sitting on the shelf in some English lab, and used it for I think it was leprosy, to begin with. And now they were sold for $90 billion. Well, obviously, small biotech is where it all begins, and it's happening the same way with myeloma, with 6 out of 8 being small biotech. So I don't want to see you guys go away. I want to see you all succeed. Get your drugs to market and give us and children a new last chance at life. So thank you.
Thank you very much, Gary.